Welcome to The Explainer. Today, we're basically putting on our hard hats and getting into some biological construction. We're going to see how these simple little sugar bricks get snapped together to build some of the most essential and complex structures in all of life.
Okay, let's dive right in. So, you look at these three: table sugar, milk sugar, malt sugar. And on the surface, they seem pretty similar, right? They're all sweet, they all give us energy. But the secret to what makes them so unique, how our bodies use them, even how they taste, it all comes down to this one tiny, incredibly powerful connection. And that connection, that special molecular glue, is called the glycosidic bond. If we really want to understand how we get from a single sugar molecule to a massive biological skyscraper, we have to start right here. This is the bond that holds everything together.
So, what is it exactly? Well, you can think of a glycosidic bond as the fundamental snap that links one sugar unit to another. It's kind of like a biological Lego connector. It's what allows us to build bigger and more intricate things from these simple single pieces.
Now, what's really cool is how that snap actually happens. What we're looking at here is the creation of sucrose, regular old table sugar. You have a glucose molecule and a fructose molecule. They come together, a little water molecule pops out, and boom, they are officially linked by a glycosidic bond.
Okay, so now that we have this amazing connector, what can we build with it? Let's start simple. What happens when we just link two sugar units together? Well, we get what are called disaccharides.
And here they are, some very familiar faces: sucrose, lactose, and maltose. Every single one of them is a classic example of a disaccharide. It's just two simple sugars joined together by that all-important glycosidic bond we just talked about.
So does it really matter how they connect? Oh, absolutely. It's not just that they connect, it's the way they connect. A really subtle difference in where that glycosidic bond forms has huge chemical consequences, and it actually splits all disaccharides into two totally different categories.
And it all comes down to this one specific part of the sugar molecule called the anomeric carbon. So if one of these carbons is left free, kind of like an open end, after the bond forms, it's able to react with other molecules. That's why we call it a reducing sugar. But if both of those special carbons are locked up tight in the bond, it can't react that way. So we call it non-reducing.
And this picture just makes it super clear. Take a look at maltose and lactose here. The glycosidic bond is formed, but see how it leaves one end of the molecule chemically open for business, so to speak? That free anomeric carbon is exactly what makes them reducing sugars.
But now look at sucrose. This is a completely different story. Here, the bond forms between the anomeric carbon of glucose and the anomeric carbon of fructose. Both of those key connection points are locked into the bond. There are no free ends, and that is what makes sucrose a non-reducing sugar.
So, we've seen what happens with pairs, but what if we don't stop it too? What if we use these glycosidic bonds to link hundreds or even thousands of sugar units into these enormous, sprawling chains?
Well, that's when you get these incredible megamolecules called polysaccharides, or sometimes glycans. These are the true polymers of the carbohydrate world. They form the massive structures that are essential for life, everything from the starch in a potato to the cellulose that makes up a tree.
And these giants basically come in two main flavors. You've got homopolysaccharides, which are built from just one type of sugar brick, repeated over and over and over again. Think of starch, which is really just a long chain of glucose. And then you have heteropolysaccharides, which are built from a mix of different types of sugar bricks.
These heteropolysaccharides, things like agarose or hyaluronic acid, are just fascinating because they're built from different units, sometimes even including things called amino sugars. Their structures get incredibly complex, and that complexity lets them do really specialized jobs in our bodies, from acting as anticoagulants to being lubricants in our joints.
And this is where things get really wild and truly three-dimensional. See, because each sugar unit has multiple spots where a bond can form, these chains don't have to be just simple straight lines. They can actually branch out, kind of like a tree, creating these vast, complex architectures with an incredible amount of information packed into their structure.
Okay, so we've gone from a single bond all the way to these sprawling molecular skyscrapers. But what are they actually for? Why does biology go to all this trouble to build them in the first place?
Well, here's the payoff. It's all about structure defining function. Those long branch chains of glucose like starch and glycogen, they're perfect for storing a ton of energy in a really compact space. Those rigid linear chains of cellulose provide the structural backbone for the entire plant kingdom. And complex chains like hyaluronic acid form protective, cushioning coats around our cells.
It's pretty incredible when you think about it. Just one simple type of connection, that glycosidic bond, when you repeat it and vary it, can create materials as different as an energy gel, the trunk of a massive tree, and the protective coating of a cell. It's an amazing example of molecular architecture. Which kind of leaves us with a final thought to chew on. What other simple connections, just repeated over and over, truly define the world around us?